Bismillahirrahmanirrahim. Welcome back friends, welcome back to another video lecture by Kami microbiologist. Today in this video lecture we are going to talk about RBC count test. In the previous videos lecture we talked about TLC, DLC. We talked their determination, that how we determine it, how we determine DLC. Now again the RBC can also be determined using hematology analyzer machine. But if let's suppose we have no hematology analyzer machine, or let's suppose if we not afford it because it's really expensive, and if we want to avoid a false report, so then what we will do, we have to count the RBC by manually. Okay, now how we count the RBC manually we will talk in this video. First of all, RBC count test. The RBC count test is actually, it is the test in which we mostly count the red blood cell in our body. Means we mostly see the quantity and the number of the RBC in our body. What are the purpose of this test? The main purpose of this test to see different kinds of anemia, or to see different kinds of RBC or hemoglobin level data decreases in this case. In this case we also check the bone marrow diseases as well. In this test we can also determine the kidney blood vessel damages. In this test we also see the morphology or the structure of the RBC. So these are the main reason, that's why we perform the RBC test, RBC count test. The normal range of the RBC count is, in male we have 4.7 to 6.1 billion cells per microliter, and in female the ratio is 4.2 to 5.4 million cells per microliter. If we see in female the ratio is a little bit less as compared to male. It is due to the menstrual cycle. Then we have RBC. So before going to explain the test in detail, we need to know about the RBC. This is not the complete lecture of the RBC, but due to this way we can easily understand about the RBC. So RBC is actually nothing but it's a non-nucleated cells, and it means that they do not have organelles as well. Only camel have the nucleated RBC, remember guys. Then we have RBC, we also called it erythrocytes, because erythro means red and cyte means cells, so red cells. Now the red color of the RBC is due to the presence of a pigment which we called it hemoglobin. Hema means iron, globin means protein. Now if we see the number of the RBC, so RBC number are higher as compared to white blood cell and platelets. And if we see the size of the RBC, so it is smaller than white blood cell but larger than platelets. Then we have shape of the RBC. So RBC are mostly biconcave in shape. As you can see, this is the biconcave shape of the RBC. Now this is the side view of the RBC, as you can see, and this is the top view of the RBC cell. Then we have the diameter of the RBC, so it is mostly 7.2 micrometer or diameter. Now we have the thickness of the RBC, so the thickness of the RBC is 1.7 to 2.2 micrometer. The specific gravity of the RBC is 1.092 to 1.101. Then we have the average lifespan of the RBC, which is about 120 days. Now the most important function of the RBC is to transport oxygen from lungs to all part of the body, and remove carbon dioxide from all part of the body to the outside of the environment, to the outside. Then we have the parameters, means in RBC count what we mostly see. We mostly count the RBC. In RBC test we mostly count the RBC. We mostly count the hemoglobin level. We check the packed cell volume or hematocrit. We also check the mean cell volume, mean corpuscular hemoglobin or mean cell hemoglobin, mean corpuscular hemoglobin concentration or mean cell hemoglobin concentration, and we also see the red cell distribution width. So these are the parameters. Inshallah, each of the parameter we talked it in one by one in some other video lecture, but in this video we are going to talk the first parameter, that is the RBC count. Then we have the requirements of the RBC, that what we required in RBC count. In RBC count we need a blood sample in additive tube, we need a white blood cell pipette, or we need RBC pipette. RBC pipette and white blood cell pipette both are the same. Okay, we need not white blood cell dilution fluid, actually in this case we need red blood cell dilution fluid. Okay, I put the figure wrong. I put here the figure mistakenly. This is actually the red blood cell dilution fluid, so we need red blood cell dilution fluid. We need light microscope, slide, and hemocytometer slide as well. Now let's see the method, that how many methods we have to find out the RBC count. As we already talked, that RBC is also find out using hematology analyzer machine, but we can also find out the RBC using simple slide method and using hemocytometer or neubauer chamber method. In simple slide method, what we will do, first of all we have to put one drop of the blood, as you can see. Now then on this slide, then we put another slide, as you can see this is another slide. Then we have to push that slide backward, as you can see backward when the slide is pushed. So then what happened, the blood are spread, as you can see on this slide. Once the blood is spreaded, so then what we will do, we have to push the slide at forward direction, due to which the blood is migrated, and the blood is migrated and spread into the tube, and we get the structure like that. So we get the blood film. This is called what? This is called blood film. Okay, now this blood film, as we already talked, this contains the tail portion, the body portion, and the head portion. So we mostly see the blood cells at the tail portion. Now these are the slide, as you can see. And now remember guys, in this case what we will do, we not stain the RBC, because RBC is already red in color due to the pigmentation, so we not stain it. Once we get the film, blood film, so then we just put wooden oil just to visualize it, and then we examine it under the light microscope. So under the light microscope, what we found the RBC just like in this form. So as we can see, these are the RBCs. So we have to neglect the white blood cell, as you can see these are the white blood cell, and we have to neglect the platelet count. Just we have to count the RBC. This is how we can count the RBC in different field, and then we have to calculate their number by using a formula which we talked in the next slide. Now let's see the RBC counting through the method of hemocytometer. In hemocytometer, first of all we have to wrap our finger, as you can see in the diagram. Second, we have to prick our finger using sterilized needle. Once the finger is pricked, so what happened, the blood will comes out. Then this blood should be picked using RBC pipette, as you can see, up to 0.5 mark. After that, it should be immediately diluted with the RBC dilution fluid. Now once it is diluted, so what happen, white blood cell as well as microbes and other things are degraded, and it should be it is protected from the hemolysis due to this dilution factor. And then what we will do, we have to release it into the slide. As you can see, how we release it, we have to release it between the surface of the slide and cover slip. So what happened, it will be spread into the slide, as you can see. After that, we have to count it. Now in this case, how we will count the RBC? RBC, as we dilute it using RBC dilution fluid, due to this way white blood cell and platelets are degraded, only RBC are remained. So RBC will be bind at that squares, as you can see, at the mid square of the hemocytometer, as you can see. At that area we count the RBC. Now each square contain 16 more square, means this one square contains 16 more square. And when there is 16, in this case 16, in the four corner we have 16 square, and in the middle we have also 16 square. So we get almost 80 squares. Okay, and in this 80 square, what we will do, we have to count the RBC. Okay, now once we count the RBC, then how we find out the number of the RBC? First of all, we have to count the number of the RBC, then we will multiply it with volume factor and with dilution factor. Let's suppose we have RBC 400, we counted in the 80 square which we talked, okay, and then we have to multiply it with volume of the factor which is 50, and we have to multiply it with dilution factor which is 200. Now once we multiply each and everything, then we get 4 millions of the RBC, which is the normal range, okay, per microliter. Then on what condition the RBC are get increased, or what are the main causes due to which the RBC are increases, okay, how we detect means when the RBC is increases, so what happen. So remember guys, RBC is mostly increases in the case of polycythemia vera. It is actually the bone marrow cancer, okay, which can produce a large number of the RBC. Then we have cigarette. Remember guys, when we enter the smoke, so due to this way the carbon dioxide is also get internalized, and it will block the oxygen attachment to the RBC. Due to this way the RBC are increase in number. Then we have congenital heart disease, which is mostly in by birth. Due to this way heart not pumping the blood, and due to this way we can say the RBC are increasing in number. Renal cell carcinoma is actually the kidney disease, and we better know kidney contain adrenal gland which can secrete erythropoietin. Due to this way, if there is less number of the if there is high number of the erythropoietin, so what happen, high number of the RBC will be, we have high number of RBC production. Now in case of dehydration, RBC level is also increasing. Why? Because if we do not have water molecules, so what happen, RBC are more concentrated. Due to more concentration, the RBC are increasing in number. Then higher altitude means that if the air pressure are increases, let's suppose if air pressure are decreases, means oxygen level is decreases. Due to this way there is a large number of RBC increasing in our body, and it will distribute the oxygen at a normal level. That's why RBC is also increasing in higher altitude. Then we have using gentamicin as well as using methyl dopa. Gentamicin we use it against microbial infection, and methyl dopa we use it in hypersensitivity or in blood pressure disease. So using these medicine, it can also increase the RBC level. Then we have low RBC. What are the main reason due to which the RBC level are decreases, or how we can find out that the RBC level are decreases in our body? RBC level will be decreases when there is thyroid disorder, because we better know that thyroid secreted actually the hormone which can cause the bone marrow to produce large number of the RBC. So if there is less number of the hormone production, so what happen, the bone marrow will be not activated, and due to this way the RBC level is decreases. During pregnancy, RBC level is also decreases, because in normal condition women have iron as well as vitamins. Due to these two things, what happen, RBC level are normal, but in pregnancy the iron and vitamins are decreases, by which the moreover in pregnancy case the blood is also transferred to the fetus area. That's why we can say that RBC level is also decreases in pregnancy. Due to the deficiency of iron, folate, copper, vitamin B6, vitamin B12, RBC is also decreases. Multiple myeloma, it is also the blood cancer. So due to this way, what happened, the bone marrow cancer, it is actually, we can say, so due to this way RBC level is also decreases. Malnutrition, different nutrients which we needed, like vitamins we need proteins, we need irons. So if these nutrients are not given in the diet, so what happened, the RBC is also decreases. Leukemia is again, it's the blood cancer, means we have a low number of the RBC, and lymphocytes are also, means white blood cell are also decreases or increasing. That's why we can say this is also due to this way the RBC level is also decreases. Due to internal or external bleeding, let's suppose internal any organ are bleeding inside the body, get injured, or if let's suppose there is injury outside of the body, so what happened, bleeding will be occur. Due to this way, the RBC level is also decreases. Blood transfusion is also the main cause of the RBC decrease. How? Because let's suppose if we take the blood from the other person, so there is a allergic reaction occur. Due to this way, the RBC are get decreases because there is antibodies. The antibodies is produced and it will cause the rupture of the other red blood cell which we take it from the outside. Then there is some hormones inside our body like erythropoietin which can increase the RBC, but due to the deficiency of erythropoietin, RBC level is decreases. We have bone marrow failure because we better know that bone marrow is actually responsible to bone marrow is also responsible to produce RBC at a normal level. But if there is bone marrow failure, so obviously RBC level is decreases. In anemia, what happens, the RBC level is also decreases because in anemia we have irregular shape of the cell, we have low number of the RBC, and we have a low number of the hemoglobin as well. Due to this way, the RBC level is also decreases. So that's all about the RBC count, each and everything. I hope you guys get idea about that. Thanks for watching.